So to begin with, uh, even no, the sperm sometimes it will be confused, like no, uh, to which the embryologist is going to subject it for either conventional or advanced sperm preparation method before they meet their partner. So coming to the evolution of sperm preparation method, no, until 1992, before ICSI, many hundreds of studies have been done on the improvement of enhancing the sperm motility, concentration, and also the total progressive motile spermatozoa, with aim to improve fertilization rate. So if you broadly look at the evolution tree, the sperm preparation methods are mainly categorized as those which are based on centrifugation and those which are based on without centrifugation. Again, there are different methods like discontinuous gradient centrifugation and also continuous gradient centrifugation. And there are techniques like swim up and swim over or swim across, which don't need any centrifugation. So I'll be talking about with this. No, I'll be in favor of these techniques. So there are other optional techniques which are mainly based on the microscopy or even binding assay techniques, and also the membrane potential based techniques, and the techniques which are based on the taxes, and also other interventions like we have customized interventions like Hawes and laser studying materials, sperm selection, and all. So I'll be not talking about this because I strictly categorize sperm preparation methods from those out of sperm selection methods. So the ideal sperm separation technique should know what are the qualities it should possess. No, it should be very quick and easy and cost effective, and also isolate as much as motile spermatozoa as possible, and also eliminate dead spermatozoa and other cells, including mainly the leukocytes and the bacteria, and also eliminate toxic or bad to substances like decapacitation factors or ROS. And also they should allow us to process a larger volumes of ejaculates. I think conventional sperm preparation methods will address all these parameters with respect when compared to advanced sperm preparation methods. Coming to the evidence and support, which is in favor of conventional sperm preparation methods, the data available regarding the conventional sperm preparation methods is enormous. Like almost the data which is available in literature based on studies and mainly are in favor of conventional sperm preparation methods when compared to advanced sperm preparation methods. And also they're time tested and also validated for the use in the human IVF. And also these methods are highly cost effective, and they can be applied to all kinds of samples, which remains the most important thing when compared to advanced sperm preparation methods. And also they're relatively easy to perform. So if you look at the various other techniques which have been extant over the evolution of sperm preparation methods, but leaving behind the two prominent methods, which is swimmer as well as density gradient centrifugation, which remains superior when compared to all other lost techniques. So coming to the literature, one of the study that has been carried out with respect to sperm telomere length, what they've done is they compared DGC as well as swimmer procedure with respect to sperm telomere length, and they concluded that both the methods can recover a sperm population with longer sperm telomere length and better DNA integrity. As you know, longer sperm telomere length means that the chromosome integrity will be good. And yes, I've just put a few slides showing the sperm selection methods as well, because I don't know, I'm expecting my opponent, Dr. my dear friend Rahul, might mention selection methods as well. So there's one snapshot from the review, which is from the Cochrane database, where they compared hyaluron binding ICSI compared to sperm flow, and the max width compared to ICSI, and zeta sperm selection compared to ICSI. So what they conclude is, they need higher quality studies, and the data is still awaited from the ongoing studies, and whether these advanced sperm selection techniques can be recommended for use in the routine practice is still a question. And I put few papers here where they compared the max with respect to sperm preparation methods, conventional sperm preparation methods, And what they show is like uh, they're not very superior or very they don't show any big differences in terms of outcomes. And also then another thing is like there is a loss of significant number of total rapid progressive spermatozoa which are subjected to max. So ultimate thing is there is no benefit as such when compared to according to these studies. And also puts a few studies here where they compared reproductive outcomes of spermatozoa which are selected by max with respect to conventional sperm preparation methods. So what they found is there is no big difference in terms of fertilization rate as well as cleavage rate and also clinical pregnancy rate. And they conclude that based on the studies we can conclude that we need more experiments like this and also registering large number of patients, exploring the different variables involved, and also in order to validate the utility of max for the human clinical practice. And coming to the data which with respect to halogen binding assay. So here is one of the largest study where they have done recently in the year 2019, which is published in the year 2019 by Miller et al. And this group. They showed 1,387 couples that underwent the ICSI procedure with or without HA. So with HA and as well as conventional ICSI. So what they found is they show very poor capacity of high binding methods for improving ICSI, and they also say that more studies are need to identify the types of patients that could actually benefit from this procedure. And coming to the IMSI again, which is based on the morphology. So here the data says that recently they compared the study has been reported with 873 sperm samples where they analyzed with MSOM, and they say that the presence of vacuoles was not associated anything with respect to DNA fragmentation and also live birth rate. And authors also claim that vacuoles are physiological features that do not alter sperm functionality. And if you look at the other data which is available, systematic review on the literature published between 2001 and 13, so author points that they lack a lot of studies, like randomized studies, that to actually corroborate the conclusions. And with respect to regular ICSI and IMSI, even the Cochrane database, I found a review where they say that uh, the current evidence from RCTs does not support to refute the clinical use of IMSI. And also they found a very low quality evidence that IMSI might increase the chances of clinical pregnancy. And also they state that they didn't find any trials reporting on the risk of congenital abnormalities. And also they suggest that well-designed and sufficiently powered trials are still required. And with respect to microfluidics, again, they compared here with microfluidics. Various studies have been compared microfluidics with respect to conventional sperm preparation methods. Again, they found there is no significant differences in terms of fertilization, pregnancy, and live birth during ICSI between both the methods, like advanced as well as conventional sperm preparation methods. So main thing coming to the evidence-based medicine. Evidence-based medicine is like which helps us to decision making, or we strongly recommend, comes out from the large meta-analysis and systematic reviews as well as large RCTs which are in favor of conventional sperm preparation methods when compared to advanced sperm preparation methods. So what the world, according to the evidence-based literature, the world weighs little more when compared to new. Coming to the conclusion, so density gradient centrifugation of SMOP still represent a gold standard for sperm preparation in ERT. And also efforts are being made to develop more efficient techniques to select the very optimal spermatozoa for IVF or ICSI, but data says that they're still insufficient. And with whatever available data, it says that they're still controversial to conclude that they are superior to sperm density gradient centrifugation or SMOP. And also conventional sperm preparation methods promises very safe, efficient way of getting the sperms in ERT, which is also time tested and also cost effective and can be applied to all and easy to perform. I would like to put this picture again because a lot of methods have been evolved over the period, but only living beyond the superior methods of conventional sperm preparation methods where SMOP and density gradient shows there. So I would like to end this debate with Advanced sperm preparation methods still lack a good scientific evidence because of poor quality of the evidences and also not enough statistical power to detect small differences, and also the lack of generalizability and mainly lack of consensus and agreement on this. So it's not for all, and they're not gold standard and superior to conventional sperm preparation methods. Thank you.
Shall I start with my presentation?
Yes, Rahul, you can start.
Yeah. Am I audible?
Yes, yes.
Yes, yes.
This presentation is visible now. Is it visible?
Yeah, please. Yes, it is.
Okay. A very warm good afternoon to everyone. Now I'll be speaking upon the advanced methods of sperm preparation. My dear friend Sanketh has successfully blindfolded you all with his words of wisdom on conventional sperm preparation techniques. Now I'll be trying my best to reveal that blindfold of yours and showing some space on advanced sperm preparation techniques. So just to shoot, before I shoot up my presentation, I would like to congratulate everyone for the 43rd year anniversary we'll be celebrating tomorrow. Why I put up this thing that I want to add, like what we saw in early prehistoric IVF, and at present times that we have seen advancements in every aspects of ART, whether it is from the laboratory equipment devices or in the embryo culture systems. We have seen advances from bell jars to benchtops. Now we are getting into the time-lapse systems, the handmade, self-made culture medias to getting commercially available media, embryo selection, which is from morphology to time-lapse, and now into the PGT era of embryo selection, cryopreservation, and now we are talking about the AI automation in ART. But there is one zone which is still we are lacking knowledge, that is the methods of sperm preparation, and we are still debating on the conventional and the advanced methods of sperm preparation. So what does that mean? Does IVF haven't seen any advances in sperm preparation, or we couldn't took ourselves out from the routine? To answer this, in the natural process of reproduction, sperm faces a series of environmental changes from the male reproductive tract to the female reproductive tract and finally recognizing the ovum. What we do in assisted reproduction, we just uh, separate the spermatozoa on the basis of their nominal parameters like counts, motility, and morphology, and with the overuse of centrifugation. And my dear friend Sanketh, this process of centrifugation not only processes the sperms with the leukocytes and the dead sperms, it also produces large number of ROS, that is the reactive oxygen species, and ultimately damages the sperm function. These conventional methods of sperm preparation, which were introduced way back in in mid 80s, and these preparation methods are still inefficient to process testicular samples, surgically retrieved samples. They basically are restricted to the suboptimal sperm seminal parameters. They their yield is less and does not guarantee that sperms with higher fertilizing ability, they cannot be able to process severe OAT samples, complete necrospermia, higher and in the semen samples with the higher incidences of DNA fragmentation. Now, yes, as rightly said, with the advent of ICSI, there is only need of one single spermatozoa to achieve a good fertilization. Currently employed conventional sperm preparation techniques basically neglect the parameters like DNA integrity, subcellular organization, apoptotic parameters, whether the cell is alive or not. There are the clinical data has shown association of live birth rate and the DNA fragmentation in the outcomes. So therefore, there is a need to develop and implement improved sperm sorting techniques and protocols in order to separate sperms with normal DNA, lesser ROS, lesser ROS, improved ART success, and decreased the birth defects. These new sperm sorting methods basically they basically mimic just near to the like natural selectivity, and they basically are based upon motility, binding, membrane permeability, morphology, and electric charge. But we'll be majorly discussing upon microfluidics and max. So microfluidics is basically a test to separate motile spermatozoa via gravity-driven device with a hydrostatic pressures to with inlet reservoirs drove flown down a converging microfluidic channel. There are the variety of microfluidic devices are available in market and being used like since last decade, and can be used in IUI, IVF, and for even for the ICSI preparation for the male severe male factor infertilities. There was a interesting paper published in Middle East Fertility Society Journal recently in 2018. What they have discussed is they have shown a relationship of progressive motility spermatozoas with progressive motility and reduced DNA fragmentation. So this particularly means that a good motile semen sample has the means the sperms with good motility, they have the tendency to have the less DNA fragmentation, and the sperm sorting methods basically are based upon the they are the valuable options in ART, and basically eliminates the damaging effects of centrifugation. Microfluidic devices use microchannels and are based on motility and morphology, and studies have been suggested that this technique not only provides sperm with higher motility, improved morphology, less DNA damage, but also has shown lower oxidation reduction potential with the reduced DNA damage as compared to swim up and density methods of sperm preparation. But these microfluidic sperm sorting devices or this technique has also shown improved IVF success in severe male factor infertility. The other technique to go is the MAX. Basically, it is magnetic activated cell sorting. MAX is based upon the selection of spermatozoa that can separate sperms with apoptotic to non-apoptotic cells. Basically, sperms with apoptosis, they have the presence of phosphatidylserine on the plasma membrane, and what this particular annexin 5 does with the help of magnetic field, that it retains the sperm cells which are damaged or which has the apoptotic markers and elute out the annexin negative, that is the non-apoptotic cells. And with the help of this technique, there are the studies which has supported the fact that max has been an important factor or a good option with to lower the DNA damage. Apoptotic markers have the good sperm recovery with the higher sperm cryosurvival rate, and also shown evidences of good higher pregnancy rates, and even has seen better results in terms of pregnancies in male factor infertility, IVF failure, and in higher cases of DNA fragmentation. Max has also been an important agent in dealing with the infertility that is caused due to the teratozospermia, and getting good quality of blastocyst available for transfer. That this publication, this paper was published in JRJ in 2018, and there was another study that has also shown the chances of max applying together with the traditional techniques, that there is no use of using max with the conventional techniques, as it to consider the cost benefit ratio, and this technique doesn't seem to be an ideal and practical method for selection. So as my friend has rightly mentioned in his presentation, the ideal sperm preparation technique must be quick, easy, and cost effective, eliminate dead spermatozoa, other cells, not cause any kind of sperm damage or non-physiological alterations to the separated sperms, that can yield a good number of spermatozoa with good fertilizing ability. That is the main point that has to be added in the ideal preparation technique. So to conclude, I would like to comment that advanced sperm preparation technique has the potential to successfully replace the routine semen preparation methods. But for that, we need to keep our mindset ready when, where, and how these techniques are needs to be implemented and executed. Spatial expertise and knowledge is required in both the methods, whether it is a conventional or whether it is an advanced method of sperm preparation. But ultimately, what we see on the tip is the basic seminal parameters are to be selected for the methods of sperm preparation. But what is still hidden, that there are the other parameters like DNA integrity, hyperactive motility, what is basically the oxidative stress. All these parameters needs to be explored and to be balanced with both the methods of sperm preparation to select one single spermatozoa for an ideal fertilization, to get good quality embryos, and and the ultimate aim of an IVF treatment is to get good pregnancy rate. All this can be happen in just a snap if we can compile together the advanced and the conventional methods of sperm preparation. Thank you.
I think Dr. Anne is there.
Yeah, hello, and
Yeah, please
okay.
continue. You have to take care of.
Okay, thank you. Sorry for my poor web connection, and congrats to both the debaters. Dr. Sanketh, do you want to counteract your debater? Rahul, do you have some
Ma'am.
points? Yeah.
Yeah, I
Please
would
carry
like
on.
to ask one question to Dr. Rahul. Uh, so, can you show me any proof, that conventional is not good in terms of results? And also, sub question is love, like it's all good, good looks very good in paper, but they might mimic the physiological conditions, but they're not translating into results, right? When compared to large RCTs and all, like data is very much support in favor of conventional sperm preparation methods, and even the infection, right, with the microfluidics is again followed by DGC are giving superior results when compared to microfluidics alone.
Yeah. Yes. There are two clinical trials which are currently ongoing that are comparing microfluidics with routine sperm preparation, and another is the max with the routine sperm preparation. So the data is not out yet, but the thing is, with these methods, basically the sperms are separated. They might be in lower yield, but the separated sperms are having good fertilization potential. I don't think the conventional methods of sperm preparation, which uses centrifugation, they might be having a good fertilization potential. As I have already mentioned in a study,
No. But see, what evidence says is like a lot of data is available. There is too enormous data is available with
yeah,
respect to conventional sperm preparation methods, right?
yeah.
So I would like to ask one more question, like, when you're trying these extra things, when there is no, which involves extra money, extra effort, or extra, as well as extra time without any
don't
extra
think so.
benefit.
I don't think so. Extra
And this
money
is not
is
applicable.
always there involved. If you
This
are
is not
using a
applicable to all, right?
if you are using a centrifuge which is having a
Yeah.
37-degree temperature and it costs you around 2-3 lakhs, at that time you can spend that amount of but you are not
See,
able
temperature
to.
regulated centrifuges is not again generalized, right? There are only few centers are using it. There is not like people are not going for it again. Like basically, they are dependent on the normal centrifuges, right? Which are based on.
No, but you can spare time of 1.5 hour for a routine sperm preparation. But there is again on the opposite side this conventional advanced technique like microfluidic max. They are eluting out good quality sperms with the higher fertilizing ability. Then what is the use? And
Followed followed by DGC.
In the
Followed
era
by
of ICSI,
DGC.
yeah, in the era of ICSI, we are only requiring one single sperm. We doesn't require a good number of spermatozoa.
No, you are totally missing out the other crucial part where we also do regular IUA, right? So for that, is it applicable?
IUI is
whatever
a
the
complete
selection
different
method.
set. Yeah.
No, we
Sorry.
are using it for IUA as well, right?
Sorry, Rahul and Sanketh, can I interrupt because we're running short of time? Can we conclude our debate? Okay, and I just like to share my views quickly because debate is a hot topic. Conventional sperm prep against advanced sperm prep. So by practice, we know all of us, even though so many years have come, still we are on to a conventional sperm preparation yet, still with swim ups and mostly with density gradients. But I feel advanced techniques are more like an add-ons to our conventional sperm preparations. Like density gradient, mostly it can be used for all types of ejaculate samples, or it can even be used for micro PSA or microsurgical sperm aspirations to remove your RBCs so that then your ICSIs are more comfortable. And with Rahul's thing, he was more on centrifugation. I know we do quite a bit centrifugation on density gradient, but there is a controlled centrifugation rate where we don't give too much damage to your sperms. And on the conventional thing, there's one more highlight is we can still do a double selective method where you do a density gradient, then from that pellet you can do a swim up, then you can take that yield. We know it might be a lesser yield, sperm yield might be less, but still you can go ahead to do your IVF and you see that more or less it's like a functional sperm. And I just like to highlight that there is in RCT papers more on support with the conventional method because it's an established method and we have a lot of live births. But still with advanced techniques, still more RCTs, Cochrane review is still not, don't have too many papers still on it. Just one paper on MAC, a few on Pixie where they say Pixie and Dixie not much of differences. But of course with selective population, they try to say there's a decreased miscarriage rate. And in that population might be a little more good pregnancy rate. And our recent 2020 RCT paper, there's one paper where they said just straight into a ICSI dish, you have your media drops where you have a fresh ejaculate, put few drops, and in the third drop, you can get your good sperm from there, functional sperm, and just do the ICSI right there. So that is just a horizontal migration technique. So by migration technique, they say still we can get a functional sperm, which is equivalent in a female reproductive tract, what happens in the cervical mucus. And so I just like to conclude that in all individual, any sperm washing technique, each individual lab approach is needed. Still universal standardization is yet to come, not done properly yet, but still people are sticking on to conventional sperm prep mostly with density gradients, but surely add-ons like MAC can be done for increased DFI patients so that you give them a better pregnancy rate and a better live birth. So thanks for your, thank you. Thanks a lot.
Yeah.
Thank you, ma'am.
Thank you.